Welcome to the Explainer. Today, we're going to decode the visual language that scientists use to make sense of some of the most complex biological data you can imagine. We're literally going from pure noise to actual, usable knowledge. So let's just jump right into the big question. How do scientists actually do this? I mean, this is the central challenge that drives so much of modern biology and medicine. How in the world do you find that one crucial signal, that one protein that could be the key to a new drug or diagnosing a disease, when it's buried in an absolute avalanche of other information? Well, the story really starts with a problem of almost unimaginable scale. We are talking about a flood of data that it's simply impossible for any human to process on their own. Okay, to give you some perspective, think about a tool like the mass spectrometer. You can think of it like a set of incredibly precise molecular scales. It weighs the tiniest parts of our cells, the proteins, and in doing that, it just creates this absolute fire hose of information from just one single sample, millions and millions of data points. And what you're looking at right here, that is the raw data. It's called an MS1 map. Every single one of those tiny, bright spots is a potential piece of a protein. Honestly, trying to make sense of this with just your eyes is like trying to hear one person's voice in a sold-out football stadium. It's just a wall of noise. So the very first challenge is, how do we filter all that noise to find the real signal? So how do researchers even begin to climb this mountain of data, right? Where do they get their first foothold in all of this chaos? All right, first things first, you got to do a quality check. Scientists will look at a chart like this one, the total ion chromogram. It's basically the EKG, the heartbeat of the entire experiment. A nice, steady, consistent line like you see here tells them, okay, the machine was running smoothly and the data we collected is good enough to start the real analysis. Now, here's where things get really clever. Proteins are huge, complicated molecules, and trying to identify them all in one piece is, well, it's incredibly difficult. So what scientists do is they use enzymes like molecular scissors to chop those big proteins up into smaller, much more manageable fragments. And these are called peptides. And this slide right here is a perfect illustration of that moment of truth. The top graph, that's the actual signal the machine picked up for one of those peptides. The bottom graph, that's what a computer predicted the signal should look like based on a known peptide. It's like a high-tech matching game. And when they line up almost perfectly, just like you see here, bam, scientists know they've got a positive ID. Okay, so let's move on to the next stage, because this is how it all builds towards a real discovery. See, it's not quite enough just to know what proteins are there. Scientists really need to know what's changed. Before you can find any meaningful differences, let's say between a healthy cell and a cancer cell, you have to prove your measurements are consistent. This visualization, called a correlation heat map, does exactly that. Check out those bright yellow squares running right down the middle. That line shows that each sample is a near-perfect match with itself, which is great. And the squares next to it are also bright, which shows that samples in the same group are all behaving in a very similar way. That's the essential green light you need to start hunting for the real biological changes. And this is the aha moment. It's called a volcano plot. And honestly, it's basically a treasure map for biologists. Every single one of those little dots is a protein. The further a dot is to the left or right, the more its amount has changed between the samples. And the higher up it is, the more statistically significant that change is. So those orange dots you see erupting from the top, those are the potential breakthroughs. Those are the proteins that scientists get incredibly excited about and will go investigate next. You know, identifying one protein is incredibly powerful, but the ultimate goal is to understand how they all work together. Think of it like a city's power grid or its transit system. You need to see the whole map. And these two visuals help us see that big picture. Over on the left, we've got a plot that finds hidden patterns in all that data. It's like a tool that automatically sorts a huge, messy pile of proteins into these neat, colored groups based on who is behaving in a similar way. And then, on the right, you've got a network graph. This is like building a social network, but for proteins. It maps out who's talking to whom, who's connected, and it reveals the real communication pathways inside our cells. So after all this, what's the big takeaway? Why does creating these clear visuals, these scientific stories, matter so much? In the end, it really comes down to communication and teamwork. As this quote gets at, the real goal here is to let scientists from totally different fields, biologists, computer scientists, doctors, to all look at the same picture and get it. It lets them discuss their findings effectively, and just like the quote says, it turns that overwhelmingly complex data into a productive dialogue. And that's the real power here. By turning millions of chaotic data points into a clear visual story, we're basically creating a new lens to see things that were totally hidden before. It allows us to finally move beyond all that noise and focus on the knowledge. And it leaves us with the most exciting question of all. With these new ways of seeing, what amazing discoveries are out there, just waiting for us to bring them into focus?